Hello and welcome everybody to our latest webinar. This one will be presented by Dr. Michael Garbati, Senior Scientist here at Active Motif. And today he'll be talking about single-cell multiome, the open chromatin plus gene expression within the same cell. So I'll turn it over to you.
Thanks, Mason. Today, we're going to start off by introducing single-cell sequencing and compare it to bulk sequencing. And then we're going to get into the single-cell multiome workflow and go through all the steps in the wet lab and the dry lab. We'll take a look at some examples of good library traces for both the ATAC and the gene expression libraries. We'll look at the Bioinformatic QC from the 10x Genomics software called Cell Ranger. We will look at what we can do with the results. What are some applications? What can we learn from multi-omic experiments? And then lastly, I will talk about our conclusions. Okay, so starting off, introducing single-cell sequencing. Traditionally, gene expression profiles have been presented as an average of the input population. This is what is referred to as bulk sequencing. This schematic compares the type of data that you could be missing if you simply performed bulk sequencing rather than single cell. Certain cell types within your population may have very different gene expression profiles. In bulk sequencing, these are all averaged together, but single-cell sequencing reveals those subpopulations and allows identification of them within your sample. We can do this for gene expression, like in this example, and we call that single-cell RNA-seq, but we can also do this for open and closed chromatin states on a single-cell level. We call that single-cell ATAC-seq. Combining these two methods into one assay is single-cell multiome. Now, multiome uses nuclei as a starting material, and each gene expression library and each open chromatin library can be traced back to the same nucleus from which they were made. ATAC stands for assay for transposase accessible chromatin, and it's a way to identify epigenetically active loci across the genome. There are several techniques for performing single-cell sequencing. The most popular one today is the 10x Genomics Chromium platform, and that's the system I'll be speaking about here. So why single-cell? Firstly, it's to identify subpopulations of cells with different transcriptomal and epigenetic profiles within the same samples. It eliminates the need for isolation strategies like FACS or magnetic sorting. We can identify drug responders versus resistant cells or tumor cells versus normal cells within a heterogeneous population like an organ or a blood sample. And single cell also gives us the sensitivity to detect a population that represents only 1% of the total population. Next, we're going to go look at the single-cell multiome workflow. So we start the experiment with a single nucleus suspension, and we first perform ATAC tagmentation. A TN5 transposase is an adapter-loaded enzyme that cuts and adapter ligates all accessible chromatin within each nucleus. Then these individual nuclei are captured in the 10x GEMs, and GEM stands for gel bead-in emulsion. We typically target 5,000 nuclei, so one nucleus makes its way into one gel bead. Within each gem, an enzyme mix of reverse transcriptase and ligase converts mRNA into cDNA, and these cDNA and the ATAC DNA fragments are also barcoded. This generates two libraries from each sample, linking both gene expression and open chromatin profiles back to the same cell. These libraries are sequenced to an estimated read depth of 30,000 reads per nucleus. And then after demultiplexing, the 10x genomic software suite Cell Ranger is used to determine how many nuclei made it into the data set. Next, we'll look at some examples of successful libraries. So here's an example of a successful multiome attack library trace. We used a fragment analyzer called TapeStation to analyze the attack libraries here. We expect there to be three major peaks at around 200, 350, and 500 base pairs. This is the classic nucleosomal patterning that the TN5 transposase enzyme creates. For the cDNA library, we see one major peak at around 500 base pairs. Before the cDNA library prep, these cDNAs are fragmented and ligated to adapters, and then spry beads are used to select for those fragments around 500. This peak can vary in intensity, but depending on which sample you're using, but as long as one major peak is at around 500 base pairs, the library is ready to sequence. After sequencing, the next step is to look at the Cell Ranger QC output. Cell Ranger is the 10x Genomics software suite that is used to analyze single-cell data. Shown here is a QC and clustering from a successful multi-omic run. We know it's successful because we're looking for Cell Ranger to call between 3,000 and 7,000 cells. And in this example, just over 5,000 cells were called. Those cells are defined by the number of unique attack fragments and unique mRNAs per barcode. The software also calls out different populations or clusters using the attack data and using the gene expression data. In the next few slides, we'll take a look at many things that you can do with this data. So in the left, UMAP plot shows defined clusters within a sample of PBMC, which are peripheral blood mononuclear cells. So these different populations were clustered by the 10x Genomics software. Focusing in the center panel, focusing on NFE2L2 gene expression in the middle UMAP plot, it seems to be expressed across these populations. However, the attack UMAP plot on the right reveals that only the monocytes have open chromatin at NFE2L2 consensus sites. Normally, the protein produced from the NFE2L2 gene remains sequestered in the cytoplasm, but in response to oxidative stress, it will translocate to the nucleus and regulate expression of antioxidant proteins. So in these cells that have both the gene expression for NFE2L2 and the potential for its gene product, the transcription factor NFE2L2, to bind, these cells are where its expression is most important. Multiome can also define subpopulations within a certain cell type. So in the left panel, we see a gene expression UMAP plot showing different types of blood cells. We can zoom in on just the T cells, and further subpopulations can be defined by the software. Here we're looking at, in the right panel, are different types of T cells that are defined by Cell Ranger. Another application of single-cell multiome is that we can define tumor cells versus normal cells. In this example, on the left panel, MS4A1, a canonical B cell marker, is used to call out B cells. Now, the middle panel, we're focusing on BANK1 expression. BANK1 is an attenuator of BCR activation, and it's repressed in tumor cells relative to the normal B cells. Next, in the right panel, we're looking at PAX5 expression. PAX5 is required for B cell differentiation, but it's also a commonly overexpressed gene in B cell lymphomas. So the low expression of BANK1 combined with the high expression of PAX5 defines this particular population as tumor B cells, as opposed to the population that has normal expression of BANK1 with normal expression of MS4A1. So in conclusion, single-cell multiome provides single-cell gene expression and single-cell open chromatin data from the same cell. Having gene expression data combined with open chromatin data can not only show which cells express your favorite transcription factor, but also which cells have the open and active loci for it to bind. Multiome can define subpopulations within a cell type and can also identify tumor cells as distinct from normal cells. Thank you.